Good. All right. So great pleasure to introduce David. David Infant to our as our speaker this week. David is to simplify things. You did everything in Ontario. You got your PhD at Queen's. You worked with Troy during your PhD, right? He was your supervisor. And then a little stint in Europe, in Switzerland, at the ETH and Bern. So you did actually you spent the COVID in Bern.
Or no, so COVID was so I was in France in between.
Okay. Oh, you traveled during COVID.
Well, no, so I
it's a little bit, yeah. It's a bit. So after his postdocs in Montpellier and then in France and Switzerland, so he's started his position at UDEM. He's in the math department at the Universite Montreal. And so David has worked on many topics in evolutionary biology, anything that has to do with evolutionary biology. He's contributed to theory and models on these topics, from plasticity, the importance of phenotypic plasticity in resolving social conflicts. But I noticed that your first paper is actually just a pure ecology.
Yeah.
Just one paper and just then everything else is evolution. But so he worked on all topics in evolution. So if you're interested in evolutionary theory, he really has a very broad perspective on the topic. But today, I believe you're going to talk on your interest in recent years, and it's really on disease dynamics, epidemiology. So I'll do that. Thanks for coming.
Yep. Thank you. Thank you for having me. So I'll start. So one of the ways in which we think about conceptually evolutionary processes is through the analogy of a fitness landscape. So basically, to construct a fitness landscape, we imagine that we have two traits of interest. If we were thinking about a pathogen species, for example, those traits might be transmissibility. How easy is it for the pathogen to spread between different hosts? And the other trait might be the virulence, so the damage the damage the pathogen causes to the host. And then for each combination of trait values, then we have some map that assigns a fitness value, right? And from that, we could then construct a landscape like this. And the utility of this analogy is if we imagine a population that's starting at this pink circle, right? Our sort of expectation would be that the action of selection operating on this population is going to cause it to basically climb this mountain, right? And so natural selection favors genotypes with increased fitness, and natural selection often maximizes fitness. Okay. Of course, real and more interesting fitness landscapes are a lot more complicated than that simple single peak landscape. Real fitness landscapes typically are rugged and they have multiple peaks. And you can imagine that this complicates the dynamics considerably, depending upon the initial conditions and stochasticity. In this case, the population could evolve to any number of these peaks and potentially get trapped there. So rather than always ending up at the global optimum, maybe it ends up at one of the local optimums. Okay, so this ruggedness really adds an interesting aspect to the evolutionary process. Now, the ruggedness of fitness landscapes is controlled by what is referred to as epistasis, or epistasis in fitness. And this is going to be the focus of what I'm going to talk about today. So the first question is, well, I want to define what epistasis is. So epistasis, the verbal definition of epistasis is that it occurs if the fitness of a mutation depends on its genetic background. Okay? I can illustrate that with a simple example from epidemiology, because I'm going to return to sort of these types of simple examples throughout this talk. Imagine that we have a population of hosts, and these hosts can be infected or uninfected by a bacterial species of interest. And we have antibiotics that we can treat these hosts with. So we'll just imagine that we have two antibiotics. Now imagine that for this bacterial species of interest, because I'm going to be exclusively interested in the evolution of bacteria and viruses within this talk. Imagine that for this bacterial species of interest, it has the capacity to acquire two independent resistance mutations that confer resistance to either class of antibiotic. And so we can then formulate basically the bacterial genetics as a two loci model where there's two alleles at each of the loci, right? So each of the loci corresponds to resistance or sensitivity to a different drug. Because there's two diallelic loci in this model, this would correspond to a situation in which there's 4 bacterial genotypes. Okay. We can use this example then to construct an explicit fitness landscape. So just imagine this little a, little b is the sensitive strain. And so we would imagine this is probably occupying a low level of fitness on the landscape. And then I've just arbitrarily assigned fitness to the genotypes which carry a single resistance mutation. And in this example, again, arbitrarily, I've just assumed that this genotype that carries big A is fitter than the genotype that carries big B. So for example, maybe we prescribe drug A at a higher rate than drug B. If we calculate the difference between the fitness of one of these genotypes that carries a single mutation and the reference genotype, which is the sensitive genotype, right? That gives us a measure of the individual fitness contributions of each of these resistance mutations, okay? And these are typically referred to in the population genetics literature as these would correspond to selection coefficients, right? So they indicate the direction and the strength of selection on either of these resistance mutations. Okay. Now let's add the genotype big A, big B. One way in which we might suppose that the fitness of big A, big B is that it's just the additive contribution of these individual fitness effects, right? So we just take the blue and red arrow and put them together, and that gives us the fitness of big A, big B. If we do that, what that implies is that the fitness of mutation B always contributes the same amount indicated by the red arrow, independent of whether it's on the background little a or big A. And same thing with the resistance mutation big A. Right? Consequently, this is a situation in which there's no epistasis in the population. So the fitness of the double mutant is just equal to the sum of the individual contributions of the mutations. And so you might imagine this is a scenario in which the drugs are prescribed independently. Okay. Of course, it need not be true that there's no epistasis, obviously, and it need not be true that, for example, the drugs are used independently. We could imagine a situation in which some percentage of the time we prescribe both drugs in combination. And if we do that, what that would do is that's going to, if you have just a single mutation and we give you both drugs, that's going to clear the infection, right? Because one of the drugs will work against the genotype or the pathogen infection. But it won't work if you have both resistance mutations. And so we would expect, therefore, that there is some additional fitness contribution that is not captured by the sum of the blue and red arrow. And this additional fitness contribution is epistasis. In this case, it's positive epistasis because the big A, big B is fitter than would be expected based upon the sum of the individual fitness contributions. And this formula here would be how we would calculate the sign and magnitude of epistasis. Of course, we could also have the opposite scenario. So rather than prescribing the drugs in combination, you could imagine that maybe the cost of resistance increased dramatically the more resistance mutations that you carry, so increase greater than linearly. For example, maybe carrying 2 of the resistance mutations leads to cell death, right? That would be an extreme form in which carrying the 2 mutations would significantly reduce the fitness of the double mutant. And so therefore, because the purple arrow is negative, in this case, or it goes in the opposite direction, what happens is that you have negative epistasis. Okay, so this is just essentially what epistasis is. We can have no epistasis, positive epistasis, or negative epistasis. Returning to the analogy of the fitness landscapes, real fitness landscapes, sorry, I guess it's this.
Just press OK.
Yeah. So real fitness landscapes, in the previous sort of description of fitness landscapes, we were probably envisioning something that's static. So I have some map and it's fixed. No matter where the population is on this landscape, that landscape is the same. And so it's not going to change. But real fitness landscapes are dynamic. And what I mean by that is if we imagine a population starting at this circle, and this population starts climbing this little peak. As the population climbs this peak, the fitness landscape itself changes. So the fitness landscape is intrinsically dependent upon the genetic composition of the population. So we have an ecological evolutionary feedback or an epidemiological evolutionary feedback. Okay. If we have a situation in which this fitness landscape is dynamically varying, so rather than a landscape, it's maybe a seascape, what happens is that no longer is there really something for natural selection to maximize, right? because the sort of the optimum will depend upon or the composition of the population. And so this gives rise to a lot of interesting new behaviors. And so the focus of my talk is going to be on how dynamic landscapes affect epistasis and some of the evolutionary consequences. I'm going to illustrate this with example from evolutionary epidemiology. So the first point would be, how would we expect the dynamic landscape to affect epistasis? So epistasis on dynamic landscapes is time and state dependent. And so we can illustrate that with, again, this very simple model that I described before. Here I'm just going to add actual equations. So we have this population of hosts that's being treated with 1 or 2 antibiotics. And so we can divide the population of hosts into susceptible and infected hosts. Infection occurs by mass action. The infection can be cleared after some amount of time, and we're treating these individuals at some rate tau. Tau AB, for example, would be the rate at which we prescribe both drugs. And that gives rise to this simple set of ordinary differential equations. Okay. Then over top of this model, we are interested in the pathogen evolution. So we would add the genetics of interest. And in this case, it's again the situation in which we have 2 independent mutations, resistance to drug A and resistance to drug B, and so 4 bacterial genotypes. Okay. For this model, we want to calculate what epistasis is, but epistasis is defined in fitness, and so we need a measure of fitness. And so in continuous time models, like ordinary differential equations, the measure of fitness that's the most appropriate is per capita growth rate. So per capita growth rate in this case is just for the pathogen or the bacteria is just the term in the parentheses there. And so we can write down what the per capita growth rate is given this specification of the model in terms of the bacterial genetics. And it's just something along some expression along the lines of this. Here I've just added some costs to bean resistance in terms of reduction in the transmissibility of the bacteria if it carries a resistance mutation, and a reduction in the duration of carriage. So the infection is cleared at a faster rate, right? And this is just to sort of incorporate the idea that having the resistance mutation probably comes with some physiological or energetic costs. Then we could just plug these formulas for per capita growth into our expression for epistasis, and we get this expression. And the most important part of this expression is what I've highlighted in yellow, and that is that this expression is no longer fixed, right? So the epistasis is not fixed. It depends upon the availability of susceptible hosts. So if we were thinking about, for example, an epidemic or something like that, the availability of susceptible hosts is going to change over time. And consequently, that can cause a situation where the sign of epistasis changes from positive to negative or vice versa. And also the magnitude of epistasis can change. So it can become more strong or less strong, right? This is one way in which dynamic fitness landscapes affect epistasis in epidemiology. It's concentrated through the availability of susceptible hosts. The other way in which dynamic landscapes affect epistasis occurs in structured populations. So imagine that we had a vaccine and we're vaccinating individuals within this population using this vaccine. So this vaccine is against some bacteria species or a virus. And suppose that this vaccine is imperfect, which means that being vaccinated doesn't mean that you can't become infected, and it doesn't mean that you can't pass the infection on to other people. And this is true for a lot of vaccines that we have, the flu vaccine, COVID, etc. But if you have a vaccine that's imperfect, from the perspective of the pathogen, what this does is this creates 2 environments, right? The pathogen can be in the environment of unvaccinated hosts, but it can also be in the environment of the vaccinated hosts. And we would not expect the selective pressures in these 2 environments to be the same, right? What that means, though, is if we were trying to calculate per capita growth rate, we would have to average the per capita growth rate across the different environments, right? And so we would have this weighted average. So RU in this case is the per capita growth rate of the unvaccinated host, and RB is the per capita growth rate of the vaccinated host. And then I'm just multiplying it by the proportion of the infections that are found in either unvaccinated hosts or vaccinated hosts, right? But these quantities are going to depend upon time. This is just a simple case in which we have no bacterial or viral genetics, but if we added some trait of interest, and so that we added an IJ corresponding to the different genotypes. So the per capita growth rates are intrinsically dependent on their genotype. there's no reason to believe that the proportion of infections of genotype IJ that are in unvaccinated hosts is going to be the same regardless of the mutations, right? We would not expect necessarily that the proportion of unvaccinated, sorry, so for example, I rather than saying what I was going to say, for example, you could imagine if one of the mutations of interest was escape from the vaccine protections, we might imagine that that will lead to a situation in which there's a greater proportion of infections that carry the vaccine escape allele in vaccinated hosts, right? Because they're basically able to evade vaccine protections, and so they might become overrepresented in that way. But then if we imagine just calculating epistasis here, right, these weights are going to be different. So even if the RU and RV are the same across all the categories, there's going to be epistasis being generated by how the population is distributed across the different environments. This is not limited to environments. So populations can also be structured by, for example, life history stage. And if you're thinking about evolution of a within host infection, if you explicitly model two stages as virions and infected cells, right, that will be two different environments, and the same principle applies. So a mutation that affects a trait that is beneficial for virions and a mutation that affects a trait that's beneficial for infected cells can have epistasis naturally emerge just through this process. Okay, so that's basically a brief overview of how dynamic fitness landscapes affect epistasis. Of course, we would then say, well, what is the point of that? And so that's what I'm going to focus upon from here on out. So the first point is the most simple and obvious, I think, and that is that epistasis produces non-random associations between mutations. And so what I mean by that is if we think about a population, again with two diallelic loci, and we just suppose in this population we've observed a frequency of mutation A being 1/3 and the frequency of mutation B being 1/3. We could then ask, well, what is the probability that if we reach into this population, we're going to select an individual that carries both big A and big B? And sort of the naive expectation would be that these mutations are independent. And so the probability of selecting an individual that carries big A, big B is just equal to the product of the probability of drawing an individual that carries big A and the probability of drawing an individual that carries big B, right? So there's nothing acting on these mutations, and so they're essentially segregating independently. And so the frequency of big A, big B is just equal to the product here. Of course, in general, that need not be true, and we can have these non-random associations emerge. So in the second case, this is a situation in which genotypes that carry both mutations are overrepresented relative to sort of the independent expectation. And in the third case, genotypes that carry both mutations are underrepresented relative to our sort of naive expectation. So this quantity, this difference between essentially the observed frequency of genotypes that carry the two mutations of interest and the expected frequency of those genotypes is referred to as linkage disequilibrium. And at least in the pairwise case is given by this formula here. And you can construct measures of linkage disequilibrium involving any number of mutations of interest. This is important because, as our intuition would suggest, epistasis generates and maintains same sign linkage disequilibrium. And so what I mean by that is if you think about the cell, if we had positive epistasis, and we're thinking about the drug example, that means that there's an advantage to carrying both resistance mutations more than would be predicted by their individual contributions, right? And so therefore we would expect there to be an enrichment in the population given an observed level of standing genetic variation of the genotypes that carry both resistance mutations, okay? And so we might ask, well, when is this relevant? And the example that I'm going to illustrate this with is multi-drug resistance, building off the examples that I've already done. So this is the example here is multi-drug resistance in Streptococcus pneumoniae. So Streptococcus pneumoniae is primarily asymptomatically carried bacteria. So many people in this room might carry Streptococcus right now. It's typically no problem and our body just naturally clears it. Within the population of Streptococcus pneumoniae, you could subdivide the population into approximately 90 different serotypes. So a serotype here just means something that is genetically distinct in how it interacts with the adaptive immune response. So each of the different serotypes will induce a slightly different adaptive immune response. Okay. The serotypes differ in terms of their transmissibility and duration of carriage, and they're believed to be maintained in the population, given that they have these intrinsic fitness differences. What's maintaining the diversity of serotypes in the population is adaptive immunity. Most relevant for us is that some serotypes have resistance to multiple classes of antibiotics, whereas others have resistance to no antibiotics. And so this is a situation in which we have a overrepresentation of multi-drug resistance. This corresponds to positive linkage disequilibrium. If we were measuring linkage disequilibrium between the resistance mutations across the entire population, irrespective of the identity of the serotype, it would be positive linkage disequilibrium there. This is important because from a public health perspective, this is the worst possible situation, right? So if you imagine we select an individual, that individual is affected by Streptococcus pneumoniae, we want to treat that individual with antibiotics. If we treat that individual with one antibiotic and it's resistant, it's more probable that that individual is also going to be resistant to any other class of antibiotics that we use on that individual, right? So we have, based upon sort of the frequencies of resistance within the population, the fact that we have positive linkage disequilibrium means that it's more likely than would be expected that basically all treatments are going to fail, right? So positive linkage disequilibrium in this case is a very negative outcome. And so this,
Can I ask you a question? I found that the number of serotypes, 90, kind of significant. So like dynamically, how can you have this coexistence, such a large coexistence of serotypes? Is there some underlying theory that we know that allows this to be true?
So that's why there is mixed reasoning for why this occurs. You're right that it's unlikely, particularly given that we have these other intrinsic fitness differences, like you would expect naively.
But some of them would dominate and obliterate the other ones, right? Like that.
But so that's why it's believed to be the hypothesis for why there are that many serotypes is that it's related to adaptive immunity. So the serotypes are differing antigenically. So if you were infected by serotype 1, for example, you can't be infected by serotype 1 in the future, but you could be infected by serotype 2, right? But that means that if serotype 1 rises to a really high prevalence, right, it's going to be at a significant disadvantage, even if it is more transmissible and it has a longer duration of carriage. It's so the pool of available susceptible hosts for that serotype is going to be basically depleted, right? So you have negative frequency dependent selection in this case, giving essentially a rare type advantage that can then maintain diversity in the population. And that segues to exactly what we were going to do with this model. So here I'm just taking that simple model that I did before and I'm extending it slightly. So now I'm dividing the infections based upon their serotypes. I'm using the superscript X to indicate serotype X, but there's potentially 90 of these serotypes. And these serotypes have their own transmission parameters. So beta depends upon serotype potentially, and the clearance rate also depends upon serotype. The treatment rate we're assuming doesn't depend upon serotype. Then within the pool of susceptible hosts, we can sort of subdivide it into overlapping sets of hosts that are basically available for serotype X or serotype Y, right? And so the complement of SX is all the hosts in the population that would have had already been infected by serotype X and therefore have antigenic or adaptive immunity to that serotype and can't be infected in the future. Okay, and so that creates a mechanism of stabilizing selection under which all of these serotypes can coexist. Okay. So from the genetic perspective, again, I'm just going to focus upon two classes of drugs, but the argument in principle applies to more than two classes of drugs. And I'm just adding a third locus in this case corresponding to the different serotypes. Okay. What that looks like in terms of the distribution of potential genotypes within serotype X is given by this set of equations. And I've just highlighted in red that I'm attaching costs to the resistance mutations in terms of reduction in transmissibility and a reduction in the duration of carriage. But that I'm assuming that these costs are not directly dependent upon the serotype itself. So these are some simulations of where I'm generating parameter sets corresponding to this model. So along the top row, what I'm doing is I'm assuming that carrying the resistance mutation comes with costs of reduced transmission. And then there is, whereas on the bottom row, it comes with costs of reduced duration of carriage. In the first column, the serotypes vary in terms of their duration of carriage. And in the second column, the serotypes vary in terms of their transmissibility. And the blue line corresponds to the average amount of linkage disequilibrium across the simulation. So in the bottom row, we see that there is no linkage disequilibrium on average, whereas in the top row, it's positive, which is what we see in the data. So then the question becomes, well, why does this occur? If we were just thinking about the per capita growth rate of the genotype IJ within serotype X, we could write it as I've done here. And the component that is important is the availability of susceptible hosts. So if we have costs of resistance that affect transmissibility, which is the first row, right? And we have differences in terms of the available hosts for the different serotypes, which is dictated by the fact that there's variation in the transmissibility and the variation in duration of carriage. This creates a correlation in terms of the selection that's operating on the individual resistance mutations due to this, the availability of susceptible hosts. So it creates an epistatic linkage between the two. In particular, the serotypes that have a strong, a longer duration of carriage or are more transmissible are fitter than the other serotypes. And consequently, what will happen is all else being equal, they will cause more infections, right? But what that will translate to is that will deplete the pool of available susceptible hosts. So they'll also be associated with the fewest susceptible hosts available to be infected. If we have costs in terms of transmission resistance, costs of resistance that affect transmission, that will mean that those costs are effectively minimized for those serotypes, right? And so that means that those serotypes are more likely to accumulate resistance to different classes of antibiotics. This is actually sort of the worst possible outcome in this case, in the sense that the fittest serotypes, so the ones that cause the most infections, would also be the serotypes that tend to be most likely to be resistant to multiple classes of antibiotics. Okay. The second consequence of epistasis that I'm going to consider is epistasis in the speed of adaptation. So the speed of adaptation is the rate at which the frequency of a mutation changes, right? So evolution is concerned with changes in frequency. Ecology is concerned with changes in number, right? So we're always interested in what is the frequency of a quantity and how does that change? In a single locus model, we could capture the change in frequency of mutation A due to the action of selection with this differential equation. So it's basically just the product of two terms. The first term, WA, is the direct selection on mutation A. This is the term that we saw before. So this is just the difference in per capita growth rate between a genotype that carries big A mutation and the reference genotype. And then this is multiplied by the genetic variance at the locus of interest, right? So obviously you need genetic variation in order for selection to operate and in order for evolution to occur. So increasing the direct selection or increasing the genetic variance, all else being equal, will increase the rate at which adaptation occurs. In a two-locus model, things become more interesting. So here we can write the change in frequency of mutation A as basically the sum of two terms. So the first term is essentially the analogous component of what we saw in the single locus case. So it's a direct selection on mutation A multiplied by the genetic variance at the locus of interest. The one modification here is if there is epistasis, and we have, and there's a situation in which mutation A finds itself on the genetic background big B, then that mutation can cash in sort of that fitness bonus and increase its direct fitness as a result of that. The second term is more interesting and what I'm going to primarily focus on here. And this is indirect selection that's mediated through linkage disequilibrium, right? So if we have non-random associations between allele A or mutation A and mutation B, and mutation B is under selection, as mutation B changes, that's going to cause concomitant changes in the frequency of allele A, right? And so if you're familiar with population genetics, in the case that allele A was for example, neutral, then we would just refer to this as basically hitchhiking, right? So the association that allele A has with allele B is going to cause, if allele B changes dramatically, that will also cause dramatic changes in the frequency of allele A. So in this picture, then we could say, broadly speaking, that epistasis has two consequences for the speed of adaptation. The first is the straightforward effect of how it modifies the fitness of the genotype that carries both mutations. And the second effect is that epistasis generates linkage disequilibrium, right? And so that will produce non-random associations between allele A and allele B, whereby allele or mutation A can profit from direct selection on allele B to change in frequency through indirect
Can
selection.
I ask a question? I was really, this is not my area of expertise.
Yeah.
I mean, the way I see this thing is just, this is a logistic equation, essentially, at least the one that you have on top, and it's modified a little bit at the bottom here. So this term that you're calling the per capita growth rate, I mean, it's just replication rate of the virus.
Yeah.
Or the bacteria.
Yeah.
So why would a mutation, like if the replication rate is, I kind of understood it when you kind of introduced it earlier, but now I'm having a really dilemma here because that shouldn't be dependent on time, that replication rate. Because I mean, why would you have adaptation if there's no mutation? I mean, I view adaptation as a way to, so that a mutation would occur to allow the pathogen to survive.
So a mutation occurs, but then once a mutation occurs, then we're interested in, given that that mutation has occurred, so there is that amount of variation present initially, then what is the speed at which that mutation is going to go to fixation, right? So I'm not considering the mutational process. I'm just focusing upon the action of selection. So given that we have this variation, right? Imagine that we have, I'm going to do this example next, but imagine that we have a vaccine and we're vaccinating hosts, and we know that there is some mutation that arises that affects the ability of the pathogen to escape the vaccine immune response. The logical question in that case to ask would be, well, how long is it going to take before this mutation actually reaches appreciable frequency in the population, right?
Oh, I see.
Because it's a difference. If it only confers a slight benefit, right, then that direct selection is going to be small, right? And that only conferring a slight benefit could depend upon the circumstances, right, in the population dynamics. Because if it was something that increases, for example, if we were supposing that we had a mutation that increases transmissibility, right, the advantage of that mutation is going to be really dependent upon the availability of the susceptible hosts, as in the previous case. And so if the infection is circulating in the population at a really high level, there's no susceptible hosts, and so there's no real advantage of that mutation. Whereas at the beginning of an epidemic process,
because it's dependent on susceptibles. That's why. Okay. I forgot about that. Okay, I see.
So that's also why the general prediction, when you have sort of an emerging infectious disease in a population, there's really strong. We would expect in general that all the mutations just increase transmissibility, because there's tons of susceptible hosts, and so that's where you can really make hay. And then only later on in the evolutionary process, once the susceptible hosts are essentially exhausted, will we have selection for.
Sorry, I think the source of my confusion was that I always thought about that in the context of within host, but here you're not doing it within host, you're doing it in a population, and it would depend on the susceptible, which makes perfect sense now. Okay.
But I mean, you could use the same logic if it within host model, but the susceptibles is the availability of cells.
Let's see. Okay.
Okay, so the example that I'm going to use to illustrate this is pathogen evolution in response to vaccination. So as we know, we know less now, but what we previously knew, established based upon science, was that vaccines are a remarkably effective public health tool. And one of their strengths is that compared to antibiotics, vaccines tend to be comparatively evolutionarily robust, right? So whereas basically resistance has evolved in response to basically every class of antibiotics that we've discovered, it's less true that has happened with vaccines, right? Having said that, evolution response to vaccination can and does occur, and there's basically two pathways through which it occurs. The first is antigenic evolution leading to vaccine escape. So if we inoculate people against this pink strain, for example, right, then that's going to cause that a lot of selective pressure on that pink strain to change its antigens and change how it interacts with the immune system and become the blue strain, right? So it can escape the vaccine-induced immune response. And that's something that we see, for example, with influenza. That's why we have to get seasonal, we update the vaccines every year. The other way in which vaccines can promote pathogen evolution is through life history evolution. And this occurs when the vaccines are imperfect. And so here, as I mentioned, if the vaccines are imperfect, then from the perspective of the pathogen, it can experience two selective environments, that of the unvaccinated host and that of the vaccinated host, with different selective pressures. And so whereas it's possible that the pathogen has optimized its life history traits for life in unvaccinated hosts, that need not be true once it's in the vaccinated host. And the way in which this life history evolution tends to manifest itself is through changes in the virulence of the pathogen or the damage that it causes to the host. And the reason for this is basically, if you cause more damage to the host, typically that's because the pathogen within the host is replicating at a higher rate, right? So it's causing more damage to the host by using up host cells, using up resources. And it's replicating in the host at a higher rate because that increases the possibility to be transmitted between hosts, right? And so this creates a trade-off in the sense that virulence is positively correlated with transmission. So you don't want, if you're from the perspective of the pathogen obviously wants to be avirulent because it doesn't want to kill the host that it's in. But there's the trade-off is that it can't be avirulent because then it won't be able to transmit between hosts. And so this is the virulence transmission trade-off. And so what we see in certain circumstances is that vaccination with imperfect vaccines can lead to an increase in virulence. And the most well-documented case of this is Merrick's disease, which is a disease that primarily affects chickens in broiler farms. So they've developed a vaccine. But over the use of the vaccine, what has happened is Merrick's disease has evolved to be increasingly lethal as a result of this vaccination process. Okay, and so the question here is just to relate it back to the speed of adaptation. How does the joint evolution of these traits affect the speed at which pathogens adapt to vaccinations? The epidemiological model that I'm using here is also relatively straightforward. I've just highlighted the key components. So when hosts enter this population, they're vaccinated at a rate or with a probability P. I assume the vaccine lasts for the lifetime of the host, although this is not super important. The protections offered by vaccination is that it reduces the probability of infection, and then by an amount theta, and then alpha in this case is the virulence to the host. So the way in which we model the damage that infections cause to hosts in these sort of models is typically by an increase in the per capita mortality rate of the host. Okay. Then we can overlay over this model a genetic model where we have the two traits of interest. The first trait is vaccine escape. So allele E potentially comes at a cost, but it allows the strain to evade the vaccine-induced immune response. And the other locus controls life history evolution. And so we have two alleles, allele B, which is the virulent allele, which is associated with higher transmission, so higher beta, but also higher alpha. And allele A is the avirulent allele. Okay? And the one thing that I'm going to add to this model that I didn't in previously is recombination. And so effectively what you can think about recombination as doing is it's essentially shuffling alleles across different genotypes. And so if you just think about shuffling a deck of cards, what you're doing is randomizing things. And so then if you're randomizing things with recombination, what you would expect is that's going to break up non-random associations that are produced by epistasis. So recombination will, the key way in which it affects these models is it removes linkage disequilibrium, right? So it's not neutralizing the effect of epistasis in terms of how epistasis affects direct fitness, but it is neutralizing the ability of epistasis to produce indirect selection. Okay. So what does epistasis between vaccine escape and virulence look like? So in this case, epistasis has to be calculated so the per capita growth rate is across two environments, and so then it becomes complicated to calculate. But we can sort of reason what we would expect epistasis to look like, whether it be positive or negative. And to do so, just suppose that the vaccine escape is complete and cost-free, and imagine we just turn off all vaccine protections in the population. In this case, we would expect epistasis to be 0, right? Because if the vaccine escape comes with no cost and there's no vaccine protections, then variation at the vaccine escape loci is going to be neutral, okay? And you can't have epistasis because it's a fitness interaction. You can't have epistasis appear with a neutral trait. And then imagine turning on vaccine protections. At least initially, because vaccine escape is complete, that's not going to affect the per capita growth rate of strains that carry the allele E, but it will affect the strains that carry allele N. And this change will disproportionately affect the per capita growth rate of strain NV, which is the more virulent strain, for 2 reasons. First, a greater fraction of NV infections are found in vaccinated hosts, and so the vaccine protections are going to have a larger effect on their per capita growth rate, all else being equal. And the second reason is because vaccine protections multiplicatively affect transmission in this case. And so turning on the vaccine protections in that case is going to have a stronger effect on the more virulent strain. Consequently, we would expect the magnitude of change in this per capita growth rate of RNV to be larger. And since vaccines that are blocking infection will decrease the per capita growth rate of the pathogen, right? When we calculate epistasis, what we would predict is that epistasis is positive. Okay, so there is an inherent advantage to carrying both the vaccine escape and virulence allele, more than what would be expected if we have these 2 beneficial mutations in the population alone. So here I'm showing the simulations of the dynamics. So along the z-axis is the amount of linkage disequilibrium, and along the x and y-axis are the frequency of the different mutations. These circles correspond to states at which the indicated genotype is at fixation. And so we start all these simulations from the wild type strain, which is the strain that is avirulent without the vaccine escape allele. And then what happens is that the system evolves to either the state where strain EA goes to fixation or strain EB goes to fixation. Okay, and it's indicated which point you end up at is indicated by the color of the trajectory. And so this is the case with no recombination. If we add recombination to this same data set, so all I'm doing between the 2 of these things is turning on recombination, what you can see is, as expected, recombination will remove the linkage disequilibrium. So in this case, since epistasis is positive, all the linkage disequilibrium that's produced is positive, potentially speeding adaptation. In this case, with the recombination, it prevents that linkage disequilibrium from building up. If we look at what the temporal dynamics look like, so this is just this trajectory where we have a lot of positive linkage disequilibrium built up in the population. If I compare the change in the frequency of the different alleles, what you can see is that the situation in which there is no recombination, which is indicated by the solid curve, it increases at a much faster rate than the case in which there is recombination. Okay, so in this case, the indirect selective effect can have a significant effect on the speed at which pathogens adapt in the absence of recombination, or in the case of weak recombinations. Okay. The third consequence that I want to discuss is the epistasis, or sign epistasis, and predictability of evolution. So if we have one of these multi-peak landscapes, right, there are potentially different outcomes that could occur. We could imagine the population evolving to this peak, but we could also imagine the population evolving to the other peak. And this affects what we would expect to occur in the long term and our ability to actually predict what will happen. And in this circumstance, this type of epistasis is, well, it's negative epistasis, but in this case, it's also referred to as sign epistasis. And by that, I mean that a mutation can be beneficial or deleterious depending upon its genetic background, right? So to acquire, if you were at this peak, to acquire the mutation big B, that's going to actually reduce fitness, right? So it's a deleterious mutation, even though to acquire big A here is positive. Okay. So I'm going to return to the example that I previously discussed, which is the pathogen evolution response to vaccination. Vaccines don't need to just block the possibility of infection. They have a lot of other consequences in terms of viral dynamics. For example, it could reduce the possibility of forward transmission, but it can also reduce the virulence, the damage that it caused to the host. So in extreme cases, this would be like an antitoxin vaccine, where you're effectively trying to remove the damage that is caused to the host. So I'm just going to modify this model here. So I removed the vaccine protections affecting the blockage of infection. I've just attached them to a reduction in virulence, and we have the same viral genetics as before. And in this case, we can go through the same argument. We're again going to have a greater change when we turn off vaccine protection, then turn them back on. We'll have a greater change on the per capita growth rate of the strain NV than strain NA. And since in this case, vaccines that reduce virulence are actually going to increase the per capita growth rate of the pathogen, right? You're effectively reducing the death rate of infections. This will produce negative epistasis. So here's again this plot that I've showed, a very similar plot. So in this case, there's negative epistasis, so this will build up negative linkage disequilibrium. And what happens is, in the absence of recombination, we have starting from the strain NA equilibrium. Each mutation individually is beneficial. So both the virulent mutation and the escape mutation are individually beneficial. And so what will happen is that strain EA and strain NV will increase in frequency, and that will drive an increase in the frequency of the two mutations. This dashed curve is the line along which the population is entirely composed of strain EA, strain NV. So there's no wild type, or the reference genotype, or the double mutant present. And at this point, this is a fast dynamic, and then this is essentially a slow manifold, along which what will ultimately determine what point we evolve to is which strain is fitter along this curve, right? So we have basically intense clonal interference between these two mutations. If we add, the trajectories are color coded to indicate what strain ultimately fits in the population. If we add recombination, that changes things. So I'm color coding the plot on the right using the outcome of the same trajectories on the plot on the left. And so what you can see is that some of the cyan trajectories go to the red fixed point in this case. And the reason why is because if we turn on linkage, if we turn on recombination, it prevents linkage disequilibrium from building up in the population. And so the population can't evolve to this curve where it's basically just competition between the two competing mutations. Okay. And so instead what happens is that there becomes an advantage to essentially entering into the population first rather than being fitter. So there's an evolutionary biostability where a mutation that arises first is more likely to go to fixation, even if it's less fit than another mutation that arises in the population while it's still segregating. Okay? And so this can affect the predictability of the evolutionary dynamics. Okay. The fourth and final thing that I'm going to discuss is epistasis and evolutionary diversification. So if we were thinking about a multi-peak landscape, we could also imagine that with a multi-peak landscape that we could potentially have genotypic specialization, where a population basically splits, and so it occupies the two peaks in the landscape. So you have, in this case, if we had the fitness of a genotype big A, big B, and little a, little b are fitter than the other two genotypes, we could imagine maybe given a certain amount of allelic variation within the population, those two genotypes will become overrepresented. So we could have just two of the possible four genotypes observable in the population. And the case on the right is the opposite, right? And so these two scenarios are characterized by an extreme amount of linkage disequilibrium. In this case, it would be positive, and in this case, it would be negative. But this linkage disequilibrium is basically maximized in both these cases. And the reason is straightforward to understand, right? Obviously, in both of these cases, we have very strong non-random associations between mutations, and so we would expect this to occur. Of course, for static landscapes, this type of diversification is unlikely because we wouldn't expect necessarily that the allelic variation within the population is going to be maintained, right? But if we have a situation in which the allelic variation is maintained by negative frequency dependent selection, right? If we then have epistasis that gets turned on, so we have this certain amount of allelic variation given by the frequencies of the individual alleles. If we then turn on positive or negative epistasis, right, that can cause the linkage disequilibrium to increase or decrease depending on whether the epistasis is positive or negative. And that will cause, for the exact same amount of allelic diversity, this type of genotypic specialization. And so the key example that I want to discuss for this is adaptive immunity and its influence on pathogen evolution. So when we're thinking about pathogen evolution in terms of adaptive immunity, typically what we think about is that we have a population of hosts. There's different bacterial or viral strains that are circulating in this population, indicated by the colors. Right? In this case, I'm saying the strains correspond to different genotypes, and where we have potentially different loci corresponding to epitopes. So basically molecules that are being recognized by the host immune system. And then there's different variants, epitopic variants at the loci, and those are corresponding to the alleles, right? After the host is infected by a strain, it clears that infection, right? It's then going to have some memory of what infected it, and that will translate to some degree of cross protection in terms of immune protections against future infection by that strain, but also by strains that are similar, and where similarity is measured by sort of genetic distance between the two strains in terms of these antigenic molecules, right? We would expect, broadly speaking, that if two strains are more similar, then it's more likely that infection by one strain is going to prevent infection by the other, right? And so that creates a strong effect of negative frequency dependent selection at the antigenic level that can maintain antigenic diversity within these populations. This is obviously critically important for a variety of reasons, for developing vaccines, for understanding epidemic dynamics, and so it's been extensively studied. What is poorly understood, what I'm going to address right now, is what can lead to sort of a subset of strains appearing in the population, right? So if we think about something like dengue obviously consists of a ton of genetic variation, but there's basically 4 distinct strains within the population, and so it's not well understood. Well, why would that occur? And it's based upon everything that I've been saying. It's probably generated by epistasis, right? Epistasis is creating non-random associations between the different antigens, which will produce a linkage disequilibrium, which will translate to one of these outcomes where we have, given an amount of allelic variation, we'll have a very specific amount of genotypic variation. And so this corresponds to a loss of linkage disequilibrium. I'm going to refer to this overrepresentation of a subset of strains or subset of genotypes as pathogen strain structure. And the other sort of open question that I'm going to also address is what causes evolutionary fluctuations in what strains are overrepresented. So evolutionary fluctuations in linkage disequilibrium. To address that, we use a sort of a standard SIR model. It's a bit complicated mathematically, but the key point with this model is that once an infection is cleared, the recovered host has some degree of immunity to the infected strain and its antigens, and this translates to some degree of cross protection against other strains that are circulating in the population based upon the genetic similarity. For the pathogen genetics, I can illustrate the key points with the simplest model of pathogen genetics, where we just have 2 diallelic loci and 4 strains. If we assume that each antigen induces a similar immune response, then by symmetry, what will happen is that there's basically 2 time scales in this model: a fast time scale over which negative frequency dependent selection will push antigen frequencies to equal abundance, and then a slow time scale along which linkage disequilibrium evolves according to this equation, right? Linkage disequilibrium in this case corresponds exactly to the overrepresentation of a subset of strains, okay? And so from this equation, what's clear is that if linkage disequilibrium and epistasis share sign, that will maintain stably a subset of strains within the population. Whereas if linkage disequilibrium varies in sign, that can cause fluctuations in the sign of linkage disequilibrium. And so fluctuations in what strains are overrepresented. Of course, the real question is, well, then what is epistasis? So in the simplest case, epistasis can be written as the product of 2 terms. The first term is A of t, which is this is a composite variable that describes the immune histories of the hosts that are circulating in the population. And what it does is capture the distribution of antigenic immunity in the population. In particular, if AFT and DFT is linkage disequilibrium share sign, then that would imply that more uninfected hosts have immunity to the overrepresented strains. And in fact, this is what we would expect. If a strain set is overrepresented in the population, it's going to cause more infections. There's going to be more immunity to it. This other parameter, sigma, or this other quantity, sigma, is a parameter. And it's effectively what sigma does is it controls the specificity and the shape of the cross-protective immune response. And so along the y-axis is just the amount of immunity a strain would have, and along the x-axis is based upon the number of antigens that are recognized by the immune system. And so sigma will be positive if recognizing a single antigen is sufficient to induce a strongly protective immune response, and it will be negative if it induces a weak immune response. Okay, based upon the fact that we expect AFT to share sign with linkage disequilibrium, what we would then intuitively expect is that if sigma is positive, then epistasis will share sign with linkage disequilibrium, and so stable strain structure would emerge. This is a bifurcation diagram that just confirms that intuition. So when sigma is negative, all the strains are present at equal frequency in this case. There's no overrepresentation of strains. Whereas once sigma becomes positive, in this case, then we have two strains going to fixation. This is stable strain structure. Okay. So this basically, and so this explains when this strain structure can emerge. It's that a subset of antigens induce a strongly protective immune response. Right. So that's the sufficient criteria to have this population where there's a given amount of antigenic variation that it becomes genotypically specialized. Of course, it doesn't provide any explanation for when and why evolutionary oscillations would occur. And the reason is because actually that model was simplified in the sense that we just supposed that it was the immune cross protection just based upon how many antigens you recognize and not what was the genetic background that the antigen found itself on. Right? You could imagine that if you were infected by a strain, that might provide stronger future protection against reinfection by that strain, right? That's not captured by just being infected by different strains that basically share the subset of antigens that make up that strain, right? So this would be referred to as homologous immunity. And so in the previous model, there was no homologous immunity in the sense that homologous immunity and full antigenic immunity were equivalent. But so if we allow for the possibility that homologous immunity can differ from antigenic immunity, this would introduce this second parameter, epsilon, into our model. And epsilon just measures the strength to which homologous immunity is more protective than full antigenic immunity. Okay. In this case, if we write out epistasis for this model, we get epistasis being the sum of two terms. So the first term is, again, the term corresponding to this antigenic immunity, which is what drives potentially if sigma is positive, that's what drives the possibility that we can have a set of strains being overrepresented in the population. The second term emerges through this homologous immunity. So SFT in this case is analogous to AFT in the sense that it captures the distribution of homologous immunity in the population, and it will share sign with linkage disequilibrium when more uninfected hosts have homologous immunity to the overrepresented strains. And this is, of course, what we expect. And so we would expect SFT and AFT, and in fact, DFT to share sign. But SFT and AFT are not the same. And so consequently, what happens is that even if sigma is positive, so there is, through the antigenic immunity, there's pressure to form this genotypic specialization, the effect of homologous immunity is to counteract that, right? And so to basically favor rare genotypes in the population. And so consequently, what happens, so here's another bifurcation diagram where I'm using both epsilon and sigma. So the previous bifurcation diagram was just along this axis, okay? And now what you can see is that there's a range of different outcomes that occur. When sigma is less than 0, then there's nothing that promotes the formation of genotypic specialization. And so we always have a situation in which all the strains are present in the population at their expected frequencies. As sigma becomes positive, it becomes increasingly likely, or the strength of a selection that favors genotypic specialization increases, particularly as epsilon decreases, right? Because epsilon is basically the force that's counteracting the effect of sigma, right? And so what happens is that we have this range of possible outcomes that can occur for the black and blue circles that include the possibility of oscillatory pathogen strain structure. So what strains are overrepresented in the population changes through time. Now, the reason why these circles or all three of these possibilities can occur is because epistasis, sigma, here I'm just varying sigma and epsilon, right? But AFT and SFT, because they are basically dynamic quantities, they will depend upon other attributes of the pathogen life history, like R naught, and whether the infection is chronic or acute. And so that will affect those, as we manipulate these other epidemiological parameters, that will affect sort of the dynamic landscape corresponding to the sort of the distribution of immunity within the population. And so I can just illustrate this by considering the star. So as I think I mentioned, AFT and SFT need not be the same, and they differ in the sense that AFT depends only on hosts that have been infected a single time, whereas SFT depends upon hosts that have been infected a single time or more than a single time, right? So there's not a reason that they would necessarily agree. For this star thing, you can see that the sigma parameter is positive, and it's also larger than epsilon, right? And so then what I'm doing here is a bifurcation diagram where I'm changing the two epidemiological parameters that are most important. Along the y-axis is the length of infection relative to the host lifespan. So we vary from an acute infection, so something like flu, to a chronic infection, so something like HIV. And along the x-axis is the R naught, so the expected, basically the reproductive number of the strain. So a highly transmissible strain would have a higher R naught. Okay. Based upon the logic that EFT only depends upon hosts that have been infected a single time, whereas SFT depends on hosts that have been infected at least once. And from inspection of this quantity, right, if we have infections being chronic, then that means that in general, hosts will have been infected maximally one time, right? And consequently, AFT and SFT will be the same. And since sigma is positive and greater than epsilon, epistasis will share the same sign at linkage to this equilibrium. And therefore, we would expect that stable strain structure to emerge, which is in fact what we occur. If we have a small R naught, then again, in this case, because R naught is so small, typically during the lifespan of a host, it will have only been infected a single time because the pathogen isn't sufficiently transmissible to infect hosts multiple times. And consequently, we would expect AFT and SFT to be approximately the same. For the same reason, then we would expect stable strain structure to emerge. And at the other extreme, R naught is really high. And so in this case, very few hosts have been infected only a single time, and overwhelming majority of hosts have been infected multiple times. So this causes AFT to go to 0. And so the epistatic term here, it becomes only the second term, which is always going to break up linkage disequilibrium. And so in this case, we have no strain structure. And so all the genotypes are equally abundant within the population. So only within this sort of intermediate region where we have acute infections that are not too transmissible can we have this oscillatory pathogen strain structure. Okay, and so epistasis provides a useful mechanism through which we can understand in considerable detail what's actually going on in the systems. Okay, so this is just to conclude. So epistasis on dynamic landscapes is time and state dependent. This has implications for the evolutionary dynamics. It can produce non-random associations, can alter speed of evolution, it can alter the predictability of evolution, and it can lead to genotypic diversification and specialization. That's it.
That's a good question. Again, I'm not an expert, so most of my questions are really naive. I remember there's a paper on dengue modeling diversity in the dengue virus, and these 2, 3, 4 different serotypes that you alluded to earlier. In that paper, they kind of argue that diversity is a result of this immunological distance between like how different these serotypes are from each other and the mortality rate. So so you mentioned you had this, you repeated online a couple of times, is that if you're infected by a different strain, you still have resistance from the previous infection from a initial strain that you've been infected with. But I thought with dengue, you have this.
So dengue is
you might actually have higher mortality
Yeah,
if you're infected for a second time with a different virus, and they argue that increased mortality is driving that diversity.
Yeah, so dengue is a special case in which you're right that basically there's I think they refer to it as original antigenic sin. So the strain that you're first infected by basically sets your immune response, and then that causes problems when antigenically similar but not. There's basically conflict, right? Where
they have to be distinct, really distinct from each other.
It confuses the immune system. So this is a good point. So actually for these ones, I did not use the original antigenic sin in terms of, because what that would I mean, it's not exactly true with something like this, but what you would predict immunity in this case would be basically maybe not sterilizing immunity in terms of a reduction in infection, but it would be instead something like your ability to survive the infection. This red circle, in fact, would be higher. So it wouldn't. If you're in this, if you've never seen the strain before, right, you have no immunity. If you've seen the strain before, you have strong immunity. But then so I guess that doesn't work with this one because I'm thinking about the amount of virulence that it would cause. But so there would be an amount of virulence that it would cause, and then for the intermediate strains in between, there would be this peak. So I think that there is, with dengue anyway, I think there is a lot of assumptions that go into the models that maybe not
convincing.
Yeah.
No.
Yeah.
Let's see.
Question. I don't want to have to go to pizza very soon, but there's just one thing that I missed that's bugging me. So in the first results that you showed, you were showing a distribution of disequilibrium, like a proportion of simulations leading to a certain disequilibrium. But I thought it was from like deterministic odds. I'm just wondering how you got a distribution of disequilibrium.
Like in this case, so I'm varying the parameters.
Okay.
So I'm varying a bunch of parameters. So this was actually I wasn't going to go into details. So this is
parameter perturbation. I had this exactly the same question.
No. So basically what we were doing. So originally there was a paper that Sonia, this Leitner paper, Sonia's paper, and we were looking. So they proposed a specific. They argued that the reason why you see multi-drug resistance is because of the duration of carriage, and they said that they argued that the logic behind that was that a longer duration of carriage means you're more likely to be exposed to antibiotics. And so what we did was this was part of this paper was part of it wasn't focused specifically on Sonia's paper, but this was an example. Was these simulations were basically using what they did, but modifying it slightly to show that, well, actually what's happening is that you have cost resistance, and then it's cost of resistance affecting transmissibility. And then if you have anything that changes the availability of susceptible hosts between the different serotypes, that will induce a correlation with selective pressures. Okay. But yeah, so I think that in the simulation there's differences in the transmission rate and things like that, and so that's yeah.
Okay. Is there any question online?
Oh yeah, that's true. Sorry. Any questions online? I don't know if they can hear me actually. Oh yeah, they can hear me. No.
Well, thank you, David.
Right.